Thanks first the organizer for having this wonderful meeting. It's really important to see all of you. So I think the question of heart regeneration, at least in mammals, at least in animals, it's not a question anymore. That we can really repair the heart, we can regenerate the heart of animals, and obviously the challenge of all of us is to somehow being able to translate that to the clinic, and we have heard the steps towards clinical translation. So these are basically our favorite molecules, processes, and you can actually see agrine molecules and the ERB2, neuregulin network. And today I'm going to focus on this ERB2 signaling in heart development repair, specifically focusing on the question of, we have many ways to restore myocardium, basically, after injury.
Can you stand behind the display?
No, it's not.
Recording you.
No, it's okay. It's okay. Stephanie was okay here, so I'm good as well. I want to move. All right. So stem cell therapy, and one of the ways that we have been trying to achieve myocardial restoration is by the de-differentiation of adult cardiomyocytes. So therefore, going backward, they proliferate, and then they somehow redifferentiate and make new cardiomyocytes. And so I'm going to speak about that. And for that, we are using a very potent mitogen. It's called this receptor tyrosine kinase, ERB2, which is very well known for heart growth, cardiomyocyte proliferation during embryonic development. Essentially, the level of these receptors downregulates after birth, but we could use this genetic tool in which we activate the receptor. We're taking a constitutively active form of the receptor, express it in cardiomyocyte, and this is a very potent inducer of growth signaling, essentially. And so we could actually show that when we use a transient activation of ERB2 signaling in acute model of MI, or in a heart failure model, we can induce very robust regeneration of the heart. So let me just focusing a little bit on our recent study on a model of heart failure. In this case, we are doing an MI in adult animal, but then we are waiting about roughly 3.5 weeks until we turn on the ERB2 signaling for roughly 2.5 weeks. These are the window, the green window here. And then we actually essentially doing we stop ERB2 signaling and we follow the heart parameter. And you can actually see that restoration of heart function was completed, and basically there is much less scarring of the heart. And this led us to suggest that ERB2 induction of transient ERB2 signaling is sufficient to restore heart function and cardiac regeneration. Looking into the molecular pathways, we found that ERB2 activate YAP signaling very, very strongly, mediated by ERK signaling. We also showed a very interesting EMT-like processes of cardiomyocytes when they are de-differentiated. Essentially, they lose connectivity, they start to migrate, they secrete enzymes that degrade the matrix, and obviously they are very much proliferative. So, but this is really important to remember that de-differentiated cardiomyocyte is not a good thing for the heart. Essentially, if we leave the ERB2 on at embryonic, juvenile, or even at the adult heart, eventually the animal will die. So de-differentiated cardiomyocytes is good for regeneration, but bad for function. I mean, evidence for that we can see from G. Martin's work, Mauro work, Thomas Brown and other people have shown the same thing. So essentially you need to make sure that de-differentiation stops, and it's a short and transient signal. Now, if you look here on this, you can actually appreciate the problem. So this is a dead differentiated heart in the mouse. This is troponin staining, very bad shape. Essentially, we lose a lot of the troponin, there is dilation. Essentially, it has to go back to a redifferentiated phenotype. Therefore, I'm saying that redifferentiation, the step that drive cardiomyocyte maturation and redifferentiation following this dedifferentiation is an important question which we decided to address. First thing we show that we ask a very simple question: what happened if we keep the RB2 on, persistent RB2 on? As I said, this is not good, and you can actually see that. So this is the short transient activation of RB2 here in yellow. So we're only focusing for simplicity on relative stroke volume, because this is showing you after MI, stroke volume is reduced during the dedifferentiation, but when we stop RB2 signaling, you start to see the climbing of stroke volume to tell you that basically the function is restored. Obviously, if you keep RB2 on, there is no restoration of heart function. And the nice thing about this system is that we don't really need to make the injury. It's a genetic system. We can just put every RB2 on, stroke volume goes down. You stop RB2 signaling, stroke volume is restored, which allows us now to zoom in what happened during the dedifferentiation, this point here, the intermediate, or during the redifferentiation. And I want you to remind that 1 month post-dedifferentiation is the redifferentiation time point. This is a bit complicated, but what I want to make sure that we did an RNA-seq and proteomic analysis. So you can see that a lot of these genes that are very much changed during the dedifferentiation, they kind of intermediate, but that eventually they go back to the same function. And by the way, there is a difference between the proteomics and transcriptomics, but the most interesting part of it is that we find that 600 genes and above 600 proteins did not go back to normal, even 1 month post-dedifferentiation. That's really tell us that something here is perhaps obscure. What goes back to normal? For example, when we look at Ki67 staining for cell cycle activity, very strongly induced by RB2, and it goes down to intermediate level and almost down to 0, but actually, even 1 month after RB2 shut off, there is still cardiomyocyte proliferation. If we go, for example, EMT-like signaling, we are using here nesting staining. Nesting is a filament of embryonic cardiomyocytes. You see it's induced by RB2, goes back. For example, mitochondrial metabolism is reduced. We see glycolysis, and this is actually restored back. But not everything restored to the same level. So we see evidence, for example, that a lot of these pathways are trying to going back to a quiescent state, but not quite there. And one of the most interesting part is this GAJA1, GAJA1 connexin 43. This is a gap junction protein. You see it's very much reduced after RB2 overexpression, but even at intermediate or even RIDIF time point, it's not restored. You can actually see it very nicely. So GAJA1 marked this intercalated disc in green here. So you see it's not restored in the RIDIF time point. Actually, we checked it 5 months or even 1 year post-RB2 activation, and GAJA1 is not there in the heart. GAJA1 is an important protein. So we actually did conduction velocity. We paced this heart. This was done by Lior Gepstein at Haifa. And you actually see that these hearts conduct much faster than the wild-type heart. Again, suggesting that something here is changed. And this led us to start to think about the possibility that perhaps we somehow, by de-differentiating the heart, we kind of rejuvenated the heart in some way. That was the suggestion that we are looking at the proteomics and single cell and transcriptomics. This suggested a rejuvenation of the heart. So we asked the following question, whether what happened if we are now doing a D-diff, re-diff cycle, meaning that we activate ERB2 in cardiomyocytes for 2 weeks, we turn it off, and then we wait 1 month after, and only now we are doing MI on these hearts. So look what happened. Now let's focus just on one thing, on ejection fraction. So in control animal, when you do ejection fraction and you do a nice cohort of animals, you will see something like that. Some animals that are hardly injured, some that are moderately injured, and some that are very strongly injured, with ejection fraction going almost to the bottom. But look what happens to these de-differentiated, redifferentiated animals. They are protected, really protected. And it's amazing. First experiment, everything is like significance. All these parameters showing protection. This echocardiography, scarring also show that they have much less scarring, showing here. So this de-differentiation, redifferentiation cycle protect the heart against ischemic injury. One of the explanations that we find for that is that this heart, these are the pre-injury. You can see that already in this heart of the D-diff, re-diff cycle, they have much more vessel in the heart. So this could be explaining why they're protected against ischemic injury. So now we are asking, okay, but most of the function of the heart is restored. So what drives this redifferentiation in the cardiomyocyte, right? So this is the question. So what we are suggesting is that the major force of redifferentiation is negative feedback loops on ERB2 signaling. The evidence for that, for example, is the HIPPO pathway. So you see that when we activate ERB2 signaling, immediately HIPPO pathway is activated in order to stop the activation of YAP, which is downstream of the ERB2. But not just the ERB2, we see a panel of negative feedback regulated. All of them are trying to stop ERB2 signaling. And by this, they try to stop. This is what pushes now back the redifferentiation phenotype. So this is seen here. So what happened now if we stop this negative feedback loop? So we are using here a very strong LATS1-2 inhibitor in culture. So you can see that when you activate ERB2, you have something like, and it's not low, about 14% of cardiomyocyte in culture. This is P7 culture, Ki67. Look what happens when you block the HIPPO pathway. On wild-type cells, you don't see anything. But when you do that on top of the ERB2, you have more than 50% proliferation of cardiomyocyte. And if you don't believe that, look at this movie. I think here. Yeah, we started to count. These are lineage trace, and this counts cell cycle activity. Not just cell cycle activity, but actual division of this culture cardiomyocyte. And there is a very strong induction of cardiomyocyte proliferation when you activate ERB2, and on top of it, you block HIPPO signaling. In vivo, we see the same, essentially showing you, so we are now generating ERB2 overexpression along with LATS1-2 knockout in cardiomyocyte. Again, when ERB2 is on and off, you see this reduction in stroke volume and then rebound of function. But when the HIPPO pathway is disactivated, in knockout of the HIPPO pathway, there is no redifferentiation. suggesting again that the hippo inhibition prevents the redifferentiation of cardiomyocyte. So this is functional and histological analysis. This is the troponin staining. You see that overexpression. Again, in the absence of the hippo, there is basically the entire heart is de-differentiated. And so there is this idea that ERB2 pushes the cells to de-differentiate the hippo pathway, try to balance that. And if you disable hippo pathway, the entire thing is going down the hill. So what can explain this protection against ischemic injury? The one explanation that we like is this more blood vessel that to begin with. There is another idea which we kind of also like, is that this reprogramming, Thomas is calling it, we call it, it's de-differentiation, redifferentiation, in a way rejuvenate the heart. And we have now heart of a young mice instead of a heart of adult mice, which is more protected. And another cool idea would be that the lack of this gap junction allows basically for a less spreading of the damage in a way throughout the heart. And again, I was just telling Jay about the story that came in neonatal pigs, that apical resection prolongs the cell cycle activity and promotes myocardial regeneration in neonatal pigs. And actually, we do see that there is 1 month after the DDIF, REDIF, we see that there is some level of cell cycle activity in cardiomyocytes. So I think this is also relevant to this model. Okay, summarize my talk here. Redifferentiation is equally important to study in the context of heart regeneration, especially if we are aiming for the clinic. What does redifferentiation look like? So functional restoration, but differentially expressed genes and protein remain. And the most important thing is that the consequence of this de-differentiation, redifferentiation is robust cardioprotection. And we suggest that negative feedback mechanism is responsible, is the driving force for redifferentiation. And just to say thanks for to the students. Gabriel Duval was the postdoc in my lab, who is now a PI in Bologna. We started the ERB2 project. Then Ala Aronov in the lab took it for the heart failure stuff. And Abby, Avraham Shaked, is the one that is working on this de-differentiation, redifferentiation. Thank you.